Bismillahirrahmanirrahim. We attempt a paper 5 and I'm going to give you the skills, the exam skills which are needed for you to attempt a paper 5. This paper is for 1 hour 15 minutes, the same time in 2022 as well, and you will be doing it on the same day as paper 2. The first thing to remember is that you read the whole question and then you read it again. So you will read the question twice and then you will start attempting it. You have 1 hour 15 minutes and this is a very long time. You have paper 2 which is also for the same time, 1 hour 15 minutes. But that is for 60 marks. While this one is only for 30 marks. Paper 5 is only for 30 marks. Now why have they given you so much time? They've given you so much time so that you can think about it and answer very, very carefully. You don't have to hurry this up and say, okay, I finished the paper in half an hour. No, you're not going to finish the paper in half an hour. You're going to spend 1 hour and 15 minutes on this paper. Let's read the question first. Some students wanted to investigate the effect of light intensity on the rate of photosynthesis in the Brazilian waterweed, Egeria densa. This plant is found in freshwater ponds. The student used the apparatus in figure 1.1 in their investigation. A stand and clamp were used to hold the apparatus in a vertical position. So they have not shown you the stand and the clamp which was used to hold the... Now what do we have here? We have a plunger of syringe. We have bubbles of gas off by the Egeria densa, or the, what was it called? It was called the water weed. So this was the water weed. This was the plant, the water weed. Not the other weed. Pond water containing sodium hydrogen carbonate. Pond water containing sodium hydrogen carbonate. Then there was a plunger of the syringe. Then there was this gas collected. So this area was where the gas was being collected. And then this is the plant here, which is, let's draw it in yellow, just to make you all realize that this is the plant and this is the length of it. It's just one stem and it's got leaves on it. And there's a rubber tubing, and to this is attached another capillary tubing. You realize why we use capillary tubes? Because even a slight movement will register. If you used a wide bore tube, then that would not really show anything. A lot of water would have to move before you would register even a movement of say 1 millimeter. So here even a little movement would result in being able to show 2 to 3 millimeters. So meniscus here, this is the meniscus here which you can see here, rubber tubing, and then there's a ruler attached to it. And let's look at this carefully and then let's look at the question what they're going to ask you. Now the question is basically pretty tough. The students carried out these steps to set up the apparatus shown in figure 1.1. Some sodium hydrogen carbonate was added to a sample of pond water as a source of carbon dioxide. Pond water must be having some bacteria, or actually the pond water in a pond would have some fish or some frogs in it or tadpoles in it which would be producing carbon dioxide. But here we are adding some chemical to it. We don't know how much chemical we have added, 1 gram, 2 gram, no. There's nothing written on it. The volume is not specified. The concentration is not specified. The syringe barrel was filled with the pond water containing sodium hydrogen carbonate. A fresh piece of E. densa was cut. The water weed was cut underwater. Why do we cut it underwater? Because so then air doesn't enter and doesn't cause an airlock. The cut stem was quickly placed in the pond water in the syringe barrel. The plunger was replaced as shown in figure 1.1 and adjusted until the meniscus in the capillary tubing was near the top of the scale. A bench lamp was used as a light source. The student noticed that bubbles of gas were given off from the E. densa. As the gas collected in the syringe barrel, the meniscus moved down the capillary tube. So the meniscus moved down the capillary tube. Why? Because as oxygen is collecting here on the top, as more and more oxygen is going to collect here, then this is going to move down. This meniscus, let me give it another color. So this fluid is going to move downwards. So the meniscus was here first, so probably moved down here. So it has moved 2 centimeter. So this is how you are going to explain this to yourself. The student noticed that bubbles of gas were given off As the gas collected in the syringe, the meniscus moved down the capillary tube. The plunger of the syringe did not move. The student used the apparatus to investigate the effect of different light intensities. So this is the thing which you are changing. So this is the independent variable on the rate of photosynthesis in the E densa. Suggest a suitable control for this investigation. Now we've done controls many times in sort of every paper we've done controls. So what would be the control in this experiment? Now in anything to do with the control, you have got to remember it's the biological organism which you are using needs to be either removed or replaced by some sort of a dead. So you could have said replace the water plant with a dead water plant of the same volume, or you can use glass beads of the same volume, or instead of pond water with sodium hydrogen carbonate you can use no water. But I mean, I would prefer you to say, just boil the organism and then use it. So replace the water plant with dead water plant. So you could have said replace the water plant with dead water plant, or you can say glass beads of the same volume, and any of this was correct. Then coming to the B part 1 of the question, describe a method using the apparatus shown in figure 1.1 that the students could use to investigate the effect of light intensity on the rate of photosynthesis. So light intensity is the independent variable and rate of photosynthesis is the dependent variable of E densa. Your method should be set out in logical order, detailed enough to allow another person to follow it. The steps carried out to set the apparatus shown in figure 1.1 should not be included. Now, if you look at the marks, this is for 7 marks. So 7 marks, and I think in the mark scheme we have about, I think there are about 9 or 11 points. So you can write any 7 of those. Yes, there are 11 points, and out of those any 7 you could have written. Now I am going to give you a very easy way of handling these. There are about 4 points which you can write in every experiment, and so in order to get those 4, you just have to give me those 4 points nearly in every planning and designing experiment, and as I go along I'll tell you which those 4 points are. First of all, let's talk about the light intensity. Now they've said to you in the experiment is that you have to effect of light intensity. So you have to change the light intensity. Now how are you going to change the light intensity? Let's talk about the independent variable. Now you have a number of choices to make. Number 1 you can say is move the distance, use the lamp at different distances from the pond. Well, that's the same thing if you said use the lamp at different power, or you can say use the lamp at different wattage, WATTAGE. So first you're going to talk about the light intensity. So you first look at what is the independent variable and tell me as much as you can about it. Maybe 1 point, maybe 2 points. You could also say use a minimum of 5 different light intensities, like, you can say 5 different distances, or you can say 5 bulbs of different power or different wattage. So you gave me first how you're going to change the light intensity, then you're going to say, okay, you do 5 different light intensity. Like if I ask you an experiment on temperature, so you take 5 different temperatures, then you say 25, 30, 35, 40, 45, that gets you another mark for telling me the different temperatures. So use a minimum of 5 different light intensities. Then use a light meter. How are you going to measure the light intensity? So use a light meter. You see many at the umpire using it on cricket matches. So you use a light meter or in any apparatus to measure the light intensity. Then important thing is, like if you're doing it in the room where you're sitting at the moment, you have more light, you have other light besides the lamp. So if you think you're in the lab and you're just using this lamp to expose this plant to the light, well then you've got to have the rest of the room in darkness, because you've got the ambient light as well. So carry out the investigation in a dark room or with no other light source. So that was another point, because if you say this because of this lamp and then you have the other lights on in the room as well, then that's not very fair. And now what are you going to measure? The dependent variable. The dependent variable is the meniscus. So you're going to record the distance the meniscus moves in say 5 minutes or 2 minutes or 3 minutes. So how far did it move? Did it move 2 centimeter or 3 centimeter in 5 minutes? So you've got to decide that how much time are you going to say? So record the distance the meniscus moves in a fixed time. This would get you another mark. So how would we word this? I'm just giving you the wordings which I would expect you to give. Use a lamp at different distances from the plant, or use a lamp with different wattages. Then use 5 different light intensities. Then use a light meter to measure the light intensity. Then carry out the experiment in a darkened room. Then record the distance moved by the meniscus in a fixed time. So this gives you 5 mark scheme points. Now let's look at this apparatus and see what are the other mark scheme points besides these 5. The first thing is use the same length of water plant. So you can see this length of the water plant. So this is the length of the water plant. It's got these leaves on it. So you could have said, what else could you have said? You could have said the same mass, you could have said the same number of leaves, or maybe even better so, the same surface area of the leaves. So if this has 10 leaves, but if the size of the leaves is different, then the surface area of the photosynthesizing part is different. So what is important is the surface area of the leaves, not the number of the leaves. So another mark for that, then another mark for the same mass and volume, same mass or same concentration and volume of the sodium hydrogen carbonate. You had added the sodium hydrogen carbonate to it, so it should have the same volume. If it is a 1% sodium hydrogen carbonate or 2% or 3%, so that is what you have to do. Then coming to the light intensity, so you would add a lamp here. I know my lamp is not a very pretty lamp. So you'd add a lamp here. Now, when you see the lamp is, you need to add something in front of it. You need to add a heat shield in front of it. Heat shield means two sheets of glass with water in between. So what is going to allow? It's going to allow light to pass, but it's not going to allow heat to pass, because if the lamp is nearer, then this is going to increase the temperature of this plant, and of course it is going to increase the rate of photosynthesis. So we don't want anything, and that is called a heat shield. You should have thought of this, that this lamp and this heat shield. So you could have thought of the heat shield. Then the important thing which you need to write nearly in every practical is few points which you can write in every practical, and one of them is equilibration time. In all the experiments you can write equilibration time. What is equilibration time? You can remember it from the word equilibrium. So equilibration time means you allow time for the water plant to acclimatize to the new environment. So the whole apparatus, the light intensity, everything needs to be in equilibrium, so that the rate of photosynthesis becomes constant. Because you pick this pond weed, this water weed out of a pond, and then put it in the lab in a beaker, and then you've set up the apparatus. So, and then you put it in this chemical sodium hydrogen carbonate, and then you put a lamp nearby it. So you've done a lot of things to really disturb it. Now you're going to put it in here and wait for 5 minutes. That is called equilibration time. So you can write that in every experiment. The other thing you can write in every experiment is repeat experiment. But here we expect you to give us a little more. Repeat experiment, how many times? So 3 times. But the important thing is 3 times at each light intensity, because you're going to change the light intensity. You are going to either move the lamp, or you're going to change the power or the wattage of the bulb. So this is, you're going to change this. So if you used a 100-watt bulb, then you do the experiment 3 times. Then you used a 150-watt bulb, then you do it 3 times. Then you used a 200-watt bulb, again do it 3 times. So repeat experiment 3 times at each light intensity. This is the most important thing. And important thing is, then you calculate a mean. So this whole thing, repeat the experiment and calculate a mean, is 1 mark scheme point. This whole thing is 1 mark scheme point. Repeat experiment 3 times at each light intensity and then calculate the mean. And the last thing which you can write in every experiment is the safety protocol. The safety protocol means that you're going to tell me about, first of all, what is the risk of the experiment. This is a low risk or a high risk. It's not a very high risk, it's a low or a medium risk experiment. So in this, what are the precautions that you're going to take? Well, the precaution is that the water weed can cause allergy. So you'd use gloves when you're handling the water weed. Then the chemical which you have used. Now this could also cause skin damage. It could burn your skin. So you use gloves and you use a mask and you use goggles, so that the chemical doesn't fall into your eyes, or but basically it's, you've got to talk about is the gloves, and the fact that you have to, health risk is from the pond water and you wear the gloves. So this is a low risk or a medium risk experiment. Uh, Right? So these are the ones, the equilibration time, repeat experiment, and the safety procedure. 3 points you can write in everything. The fourth point, which I want you to remember here, it's not sort of mentioned, but how are you going to maintain the temperature? And using a thermostatically controlled water bath to maintain the temperature is a fourth point, which you can write nearly in every experiment. Here, of course, the temperature has been attributed to the heat shield. So this is the way you maintain the temperature, so the temperature is not maintained. But if you said, in another experiment, you could have said, maintain the temperature using a thermostatically controlled water bath. Just quick recap of the wordings. Use the same water plant. Use same concentration of sodium hydrogen carbonate. Use a heat shield to reduce the heating effect of the lamp. Allow equilibration time for the water plant to each light intensity. Repeat at least 3 times at each light intensity and calculate a mean. Low risk experiment, use gloves when handling water weed and pond water. There are no marks for the chemical handling because it wasn't, I think it's not so risky, so the chemical was not given any marks. Now coming to the part 2 of the question, complete the sketch graph to predict the result that you would expect from this method you have given in B1. Include axis labels and units in your answer. So naturally, what are you going to give me? You're going to give me light intensity on the x-axis, and you're going to give me the rate of photosynthesis on the y-axis. You can also give me the, if you didn't want to give me light intensity, you can also say power of the bulb, or you can say distance from the light bulb. And in the rate of photosynthesis, you could also have given me rate of movement of the meniscus, or the distance the meniscus moved in a set time, or time taken for the meniscus to move a set distance. And the units for light would have been distance from the light bulb could have been in centimeter or millimeter. Light intensity could be in lux or candela, and power rating of the bulb could be in W, wattage. The rate of photosynthesis would be in centimeter per minute, or millimeter per minute, or centimeter per second, or millimeter per second, or you could have said centimeter per minute. So basically you're going to give me that. And then of course the graph of course would be very simple. Light intensity goes and then it levels off. Units could be very variable. Light intensity could be lux, or could be candela, or could be wattage of the bulb. If you're saying rate of photosynthesis, then the rate of photosynthesis, you have to give me a unit of time. But if you said distance the meniscus moved, then you could have just said centimeter or millimeter. Or if you had said time for the meniscus to move, then you could have said in minutes or seconds. So rate of photosynthesis is a different thing. You have to mention time. But if you said distance meniscus moved, then it could be centimeter, millimeter. And the graph shape is very common. Light intensity increases and the graph goes up, and then it levels off, becomes constant, because light intensity is no longer the limiting factor. Now coming to the C part of the question, the student decided to use paper chromatography to separate the photosynthetic pigments found in E. densa leaves. E. densa leaves were ground up in a small volume of solvent to make a concentrated solution of pigments. The different pigments in the solution were separated using paper chromatography. The distance moved by the solvent was measured on the completed chromatogram. The distance moved by the pigments were also measured. The RF values were calculated for these pigments. The student, table 1.1 shows the results. Pigment 1, 2, 3, 4, table 1.1, color of the pigment orange, yellow, blue, green, green. Distance moved by the pigment in millimeter 105. Distance moved by the solvent in millimeter 107. RF value is 0.98. Then for yellow it is 94, 107, and we have to calculate this. This is the one which has been left out for you. Blue green was 62, 107.58. Green was 45, 107, 0.42. So you have to calculate only one which you had to do. So complete table 1.1 by calculating the RF value for pigment 2. Give your answer to two significant figures. The formula for the calculation is distance moved by the pigment over distance moved by the solvent, and this is 1 mark. Then the simple maths was 94 divided by, you see it's the pigment. And this is the pigment. The pigment was 94 divided by 107 is the solvent, and this gave you 0.88. Now coming to the next part of it, it says the student used a published source to find the RF values of three of the pigments that they expected to be present. So RF of carotene was 0.95, RF of chlorophyll was 0.60, and RF chlorophyll B, this is chlorophyll A, was 0.60, and chlorophyll B was 0.50. The student correctly identified pigment 1 as carotene, pigment 3 as chlorophyll A, and pigment 4 as chlorophyll B. The calculated RF values were not exactly the same as the RF values in the published source. Describe one difficulty with paper chromatography that could explain why the RF values calculated by the students were not exactly the same as the published data. This is only for 1 mark. I mean, if you don't know it, don't spend a lot of time on it, but just make a calculated guess, and I'm sure you could get away with it. But you must sort of go in your mind the whole process of chromatography. Now, I want you to just revise this chromatography, and you can see here watch glass containing concentrated leaf extract, and then you load the sample, and then you set up the chromatography chamber, and you allow the solvent to move up, and then you see the pigment moving up in different lengths also. So chlorophyll B, chlorophyll A, and then xanthophyll, and then carotene. So you can see how they move different distances. Now, similarly, in another diagram, I want you to see another diagram. Now in this diagram again you can see the solvent line, then you can see carotene and xanthophyll and chlorophyll A and chlorophyll B, and the pigment extract is the origin. Now all these points, when you look at these points, now what was the question? Let's go back to the question now. Describe one difficulty with paper chromatography that could explain why the RF values calculated by the students were not exactly the same as the published data. Now, if you keep on looking at this experiment, and you can now understand the points that I'm going to give you. The first point is difficulty in achieving a spot of the extract. Sometimes it might not be so concentrated and just spreads out and diffuses. Then the other point which you could have given me, each pigment spreads out during migration, gives a long streak or runs into each other or overlaps. Number third point you could have given me, pigment may fade, may be too pale to see clearly, so difficult to measure accurately, as you can see in some of these. Then the fourth point you could have given me, it could be quality or the grade of the chromatography paper. Then the fifth point you could have given me was the type of solvent. Sometimes the solvent evaporates and it is improper preparation of the solvent. Then the sixth point you could have given me was the different laboratory conditions. The temperature could be different, the percentage saturation of air with the solvent could be different. And another point, seventh point, you could have given me that the chromatogram was not replicated, meaning it was not repeated. We just did one chromatogram, we didn't repeat it. So this repeat thing you can write nearly whenever you've been asked, if it's a complicated question, you can write that if only one measurement has been taken, that this was not repeated. Because if you did not repeat it, then it's not a very reliable result. So please remember, for paper 5, you have to be very smart and you have to be very careful when you word these. So you must take your time, you must think it out, you must think you're actually doing this in the lab, and you must mentally simulate that you're doing this in the lab, and then think what could be the possible points. I will finish this video here, and there are two more questions in this paper, and we will do that in the next video which follows after this, which will probably be done by tomorrow morning. Thank you once again for watching, and I hope this is helpful in helping you solve the paper 5. Thank you very much for watching. Please do subscribe. Thank you.